Hello, Dr. Shailesh Sharma. Abindya?
Yes, I can hear. Can everybody hear me? Prabhata, can you hear me? I cannot hear you. You are muted.
Yes, in India we can hear.
Yeah. Professor Mandal, you are muted.
Yeah, yes.
We'll get started in 1 minute.
Arindo, we can hear you in the auditorium, no problem.
Great, great. So, Professor Mandal, let me know when I can start. If Professor Balaram, the chief guest, is around, I can get started with the day.
I spoke to him. He said he's there. So I called him. I talked to him. So
Okay.
maybe another a minute or so. Another
Sure.
a minute. Yeah. So they can see that on the screen. Chief guest has Professor Balaram has not joined, but what shall he do?
We'll wait for him to join.
Huh?
We'll wait for him to join. Then we'll get started.
Yeah, okay. Sarah joined. Sarah joined.
Okay, great.
Okay.
Can everybody hear me?
Yeah.
All right. Good afternoon, everyone. My name is Anindya Ghosh Roy. I'm an associate professor at the National Brain Research Center. It is my pleasure and honor to welcome all my friends, colleagues, students, fellows, and all the dignitaries and the guests in today's public lecture event, which is the major event in today's 18th Foundation Day of NBRC. NBRC usually fondly celebrates Foundation Day with multiple scientific events, including scientific lectures, open day for the school students and the college students. Unfortunately, due to COVID situation for last 2 years, we are not having these activities. But today we are very honored to have a very special guest, Professor Padmanabhan Balaram, who has kindly agreed to give this public lecture. First, I would like to request Professor Mandal, our director in charge, to say a few words about NBRC and the Foundation Day. Professor Mandal.
Thank you, Anindya, for a nice introduction and introducing our beloved Professor Balaram. And it's a big honor for him. He's there with us and giving a talk. Nothing can we have type of research less than 12 minutes. So I just want to give a quick talking to. In a cool mind. They came from different parts of the countries and also trained in various leading laboratories. They left their research, left the country, United States, Germany, and in other countries, developed countries, and came to India, and NBRC joined to do the cutting edge research. They are unique. Nobody is replaceable by another person. So they are so unique. So we just start to talk about their research activities of this. So before we telling this one, this is a matrices people talk about it, that we do involve research, and that research is actually what type of research and how people see us, our quality outcome in terms of citation. So NBRC has a H index in 70 from Google Scholar. So this is we have, and then we have a program called PhD and MSc program. We are deemed university from 2002. And our accreditation process is already completed. We have some news at the end of this lecture. So this is the different variability, and we are continuing. Our research has been well cited in various cases. And NBRC is very, very unique. In this sense, we attract foreign funding. We attract top funding agency in the country, not only DBT. DBT is like our mother. But there are any other organizations, they are actually give us for our various outcome measures. And it has a various facilities unique for brain research, like a state-of-the-art NMR facility, MRI facility, you make facilities, two-photon microscopy. There are different other facilities available. Microscopes are unique in these cases. They are unique for brain research. And you can see the profiles of funding. This year, because of the funding, there was extra amount was given to us, and next year 5% increase because we are doing good from the DBT in the time of crunching. So it's a case of good satisfaction. Now we actually we get money from different organizations. That's what we survive, apart from government help. United States Air Force, NIH, Ministry of Youth Affairs, Electronics and Information Technology, so many other organizations. So this is our actually our own person. These are all students. They did so many, there are so many students, I cannot even tell you. They are actually, this is few of them pictures we got, and some of them are collection stage. So they are actually our pride. As a teacher, as a faculty, they are our actually ambassador. So right now NBRC has given 96 PhD so far. So we are sort of to get 100. That was my target in 2021. So here I talk about research component. Research component is that NBRC is like a family. All faculty members hang around together. They kind of discuss together and they build together about brain research, different aspect of it. So there are 5 sections in there, but basically there is no section, is open space. This is just a phase of NAC accreditation. We have to do this kind of arrangement. So these are the one of the work. I'm just sharing all faculties' work briefly, very briefly, because they are elaborative. This is that role of cortical interactions. Usually they are actually looking at what are the connectivities. And this work has been published in Cerebral Cortex, one of the prestigious journals. So this why it will take a little time for explaining, but these are available in this journal. Several can be downloaded. Then another aspect is that what is our contribution for this COVID time research? Our scientists actually very much in leading role, like Elora Sen has done some Madhus. They actually protect us from this COVID. This is ancient time-wise, it is there. And based on our research, original work, there is a clinical trial in Jammu. So you can see that we decide, and other people actually, some of them do the clinical trial. Then another work is on Pankaj said, he is actually looking at that COVID, we should not be looked at on the respiratory system. It has a lot to damage at the brain. So he is looking at the biological aspect. So this is very important because people are very busy for looking at the pulmonary system. So another thing is that, excuse me. So here we also, as a imaging, also another aspect of it, we said that we want to monitor the mental health by imaging. So that means some things who will be having some psychological problem, psychiatric problem, they can be monitored and we can predict it. That type of work has been done and it has been highlighted. Now, another work of Dr. Anirban, he's gone out of the way, see that how monocycline can be effective for Japanese encephalitis. His original work was actually taken to for a translational mode, and there are clinical trial has been done, and it was found out the sample size need to be done in a more number of people so that it can be tested, so that a lot of people's life can be saved because of his research. Now, another aspect of it, that we are collecting data from ourselves, publishing it. But this data is unique. There is a big initiative called Swadesh has been generated. So this Swadesh actually is a unique proposal in the world where brain neurochemical data is available, and that data can be being tested in patient and is arranged in the close collaboration with AIIMS, Dr. Manjari Tripathi. So this is an original work available in the center, published data cited extensively, and that has gone for clinical trial. So this is, you see, this data can be taken by other people, and Honorable Minister 3 weeks ago actually inaugurated this center. He kindly visited along with our secretary, new secretary. So it is a matter of pride for us. Now, this one, next one, this one. So this is, you see, not only we do the cutting edge fundamental research, we have also a clinical trial is coming up, that antioxidant glutathione, whether it can seize the impairment of the MCI patient, that is waiting for to start the process. Another also, our colleague has started, Nivedita, so she is going to do the Parkinson disease related some levodopa, how does it affect the Parkinson disease patient. So you can say 2 clinical trials already stand up, waiting for to start after COVID. Then there is I showed you the pictures. Each one was telling that they are unique. Anindra is very interested to understand how neurons are polarized in initial stage of development. And he talks very simple, but his goal is very high. And his work has been published in Proceedings of National Academy of Science. So it speaks for itself. Now, Sieb, good friend Sieb is also looking at the memory formation. And he is actually integrating this cell, and his interest in Alzheimer disease, how the memory, how it can be involved in a synaptic plasticity and memory. So this has a serious implication in biological aspect of Alzheimer disease research. Professor Ranjit is another aspect he has brought, which is actually is looking at that understand molecular mechanism between amyloid beta peptide and prion replication. So he's the only person working this National Brain Research Center and prion research. He's trained from Nobel laureate Group Prushner. He actually came from there and from this knowledge base. So to translate a prion, give a new direction. He has published a lot of good papers and many more to come. So here, this is Dr. Saurabh, and very happy to tell you that his work in cloth biology came as a front page, that cover page. And actually, this is very, very important for NBRC, not from him also. So this, he has actually RNA-based mechanism of synapse formation, synaptic plasticity and memory formation. He has many papers actually in a top journals. He's been lined up in a various stage of acceptance. So it's a matter of, he's a relatively new person and is opening up in various great directions. So this is Soumya. So Soumya is a very unique in the country. She is actually trying to investigate the behavioral response to mirror and mark test in adult zebra finches and house crows. So she is actually looking at how these animals behave and how their cortical connectivities are there. So this is another, it takes about a day to explain her detailed work. So this is very much important, and we have a lot of crows in this our NBRC center. Here is a Bhavani. This is very important. Bhavani is a new recruit, and he has opened up a new direction, specialized vesicle trafficking pathway in neuron and neurorecurring cells and roles in diseases. So he has started his lab in a very strong footing, and I'm sure he will have many more good publications and work will come out from him. Last one is the Dhruva is he's trying to understand the Drosophila model for Alzheimer disease. Very simple system, but very complicated disease. And he's setting up, he's also 1 year, he's here with us, and many more things will be coming. He has excellent model. So he's working very hard in this direction to establish. Then apart from our research, also we do a lot of social work. We help as India as our friend, and many people come to NBRC. Our scientists go to the various organizations and various school, college to brain awareness weeks. And also we have our many organizations are trying to have a collaborative MOU with us, Florey Institute, University of Pittsburgh, and then other engineering institutes. They are also trying to have MOU so that we can have a collaborative research. Also, I want to emphasize that in the COVID time, we reach out to the civil hospital, we help them with all support. So many people did their scanning time much faster then without our help. So they are like a part of our family. We help them many ways. They help us for vaccinating our student much before actually other people did. So our student, God grace, there is no casualty at NBRC, although there are some cases where they all saved. We have a fantastic RMO. He actually was living basically with them. He saved us. So I must be thankful to him for this. We have a green canopy. We are actually fill up the entire places with the trees, and that type of activities. Our students are very active. They participate with our staff member, and then we have a APJ Kalam award to encourage our students so that they can do very well in publication and leave the institute in 6 year times and go to the various places to do very good work, or they join somewhere else in India to enrich our country. So this is the slide I want to give it to you, that I would like to say that thank you very much for all support we get from DBT. We have a new secretary, scientist, friend, colleague. So that's a big thing, strength for us. We have a minister visited, and he said in his writing that we'll come back again and spend an entire day with us. He was so pleased, and we have so many supporters, friends from other DBT institutes. They support, help us, and DBT like to say with a humble things. We tried hard, work like a team, and that's a very good news for us. That NBRC was a university of in second from deemed to be university from 2002. Then in the process we learned, and then we start the process of accreditation.
Which major audience today are the students and the faculty of NBRC. So it will be block different ion channels. So here is the angle of Professor Balaram, which is connecting to the neuroscience research of our institute. Without further ado, I would like to invite Professor Balaram to give his lecture titled Chemical Behavior and Neuroscience. Professor Balaram, thank you.
Anindya, can I share my
Screen, yes. Yes,
No?
please, sir.
Yes.
we get something in?
Okay.
Can you see my screen now?
Sir,
It's
yes,
coming up.
coming up, sir.
I'll try and make it full screen.
Yes, I can
Yes.
see now the screen here.
We can see.
Just wait for a second. I've made it full screen, so you should be able to see it full screen.
Yeah, we can see it now.
Okay. Thank you. Before I begin, I must tell you that my internet connection is somewhat unstable today. So if my voice breaks, just be patient and I'll come back.
No problems.
When Dr. Mandal invited me to give this lecture at NBRC, I was a bit worried because I'm not a neuroscientist. And he said maybe I could talk about the role of chemistry in neuroscience. And I had given the title of Chemicals, Behavior, and Neuroscience. And in the email which I received this morning, I found that there was a typographical in which the letter S and the comma had been deleted. So it's what I would call a deletion mutation, which can affect phenotypes, as in the case of the coronavirus. It happens when we leave out, but when we convert plural into singular and leave out a comma. I show you a book. I made the slide just before this lecture. Very interesting book, which uses the phrase eats, shoots, and leaves. And there's a comma there. And you can think about it yourself. The punter gets up and removes the comma. And you can see two completely different meanings once the punctuation is gone. But in beginning this lecture, since I am talking to an audience of neuroscientists, I'll begin with Francis Crick. He wrote a book called The Astonishing Hypothesis in 1994, in which he said, and I take this from his obituary, which appeared in Cell just after he had died in 2004, written by Robin Holliday. He says, Crick says, you, your joys and your sorrows, your sense of personal identity and free will are no more than the behavior of a vast assembly of nerve cells. As Lewis Carroll's Alice might have phrased it, you're nothing but a pack of neurons. This hypothesis is so alien to most people who are alive today that it can truly be called astonishing. And he entitled his book The Astonishing Hypothesis in 1904. Years later, the century was ending, the new century and the new millennium were beginning. He wrote this in the millennium issue of the Philosophical Transactions of the Royal Society. He said, neuroscientists should scan molecular biology for appropriate techniques, but most important, they should ask their molecular biology friends for new tools. They should tell them what their difficulties are and what they want to do. Once the word gets around that a certain type of problem exists, it is surprising how often someone has a bright idea of how to solve it. So don't be shy, ask. After all, exactly how our brains work is of vital interest to us all. So why shilly-shally? just for the benefit of those of you who are here, because I also found this usage of shilly-shally strange. What it means is, why should you hesitate? Why should you be indecisive? Why should you delay in asking for collaboration? The effect of chemicals on the brain has been known for a long time. The most dramatic example which I had seen when I was young was when LSD, or lysergic acid diethylamide, a hallucinogenic drug, became popular among college students and university students in the United States. Lysergic acid itself is derived by the hydrolysis of a natural product called ergotamine, which is a product of a fungus which grows on rye. And you can see here where I've drawn the red line. If you break that bond, then you get lysergic acid. Albert Hofmann, in the late 1930s and in the early 1940s, working in Switzerland in a pharmaceutical company, made many, many derivatives of lysergic acid. And he made the diethylamine derivative, which is the diethylamide. And then by accident, I think he must have had a little bit on his fingers. He ingested it and he had strange feelings. He first thought it was because of the chloroform that he was using. Later on, he realized that it was from LSD, which he had discovered. Hoffman did an interesting experiment in 1943. He made a very, very small, low concentration of LSD solution and drank it. And after that, he described what he felt. And that was a remarkable description which you can find on the internet. I've given you one reference here. April 19th, 1943, he actually ingested LSD, and then he asked his assistant to take him back, both of them riding bicycles in those days. During the Second World War, everybody rode bicycles, petrol was scarce. And when he rode the bicycles, he felt enormous sensations. He was not sure he would reach home, and he had ingested an extremely small amount of LSD. So here was a chemical which had a remarkable effect on the brain. This article in Scientific American is the story of Bicycle Day, which is celebrated every year on the 19th of April. And in 2014, there was a major conference which Hoffman attended. LSD apparently had no great effect on Hoffman. He took other molecules like psilocin, which he discovered also. He lived to the age of 102. Not because of this chemical, but he just lived to the age of 100. Plants produce a number of chemicals. I show two of them here. They're what we call natural products. Caffeine on the right and morphine on the right. Coffee plant and the opium poppy. And we can ask a scientific question. Why do plants make these complex molecules? If we ask this question, we're really revisiting the chemistry of natural products. Now, it turns out that molecules are made by microorganisms, they're made by plants, they're made by animals. In a biochemistry textbook, we classify them. We break them into primary metabolites and secondary metabolites. There is nothing secondary about secondary metabolites. This is because plants invest a huge amount of genetic and metabolic energy in producing these molecules, both plants and microorganisms. I found this definition of secondary metabolism many, many years ago, which says that it represents the splendid idiosyncratic diversity of nature, endowing different species with specific solutions to biological problems. We can now ask some questions. How many chemicals are produced in nature? This is what we will call chemical space. We can ask how many living organisms are there in nature? That would be biological space. We can ask how are these chemicals synthesized? This would be the biosynthesis, which requires genes, gene clusters, regulation, and enzymes. You might also ask a more biological question. Why are these chemicals necessary for the organism? What is the biological imperative? In that case, we get into the field of chemical ecology. And of course, last of all, we can ask how are the chemicals of one organism recognized by the target organism? This takes us to the subject of receptor proteins and cell energy and information transduction in cellular systems. On this slide, I show you one natural product which you're all familiar with. The component of red chili which gives it a pungent taste, capsaicin. Capsaicin is an important molecule you will see on the next slide. It's made by this complex biosynthetic pathway. Each arrow requires an enzyme. Each enzyme requires a gene. So the plant is investing an enormous amount of biochemical metabolic effect in trying to make this molecule. It's very useful for the plant itself. There are many other molecules like this. Look at capsaicin and look at curcumin, which you find in turmeric and which everybody in India knows about. You will see that the left-hand side of these molecules look very similar. There's an aromatic ring, a phenol, with another substituent. These are derived from the smaller molecule vanillin, and they're called vanilloid-type molecules. Why are these molecules important? Because it is these molecules which have permitted the two Nobel laureates this year for medicine and physiology, David Julius and Ardem Pakutian, to actually establish the existence of the transient receptor potential on the so-called trip ion channels. And it turns out that these molecules, in order to actually identify the channels, the capsaicin molecule is what gives us the receptor which allows us to sense heat, and menthol is what is used in the receptor which allows us to sense cold. So sensory perception and signal transduction are very important for our everyday life. And since you're neuroscientists, I thought that I would take a kindergarten picture of what we teach children are the five senses. We teach them that the five senses are touch, sight, hearing, smell, and taste. That's on the left-hand side of this. On a receptor protein reactions. And on this next slide, I detail this a little bit, which responds to light. Toxin, which absorbs right in the middle of the visible spectrum, nanometers, which allow us to feel touch, and then you have molecules which allow us to smell, and also in the ear, of course, a slightly more complicated thing because you're really listening to auditory signals. It turns out that in biology, all organisms except human beings communicate exclusively by means of chemicals, very little by means of sound, and certainly not by means of language. So much of our discussions of chemistry and biology could be to discuss chemical communication in biology, chemical diversity in biology. Communication in biology can be between cells or between organisms. If they are between cells, the chemical effectors are called hormones. If they are through space, either through the air or through the soil or through water, they are then other molecules, pheromones if they are volatile, diverse chemicals which are non-volatile. So everything that you see around you in biology is the result of organisms communicating with one another by means of chemistry. Because chemistry is sometimes complicated. Why am I emphasizing chemistry in an institute for brain research? I must tell you this, that the best definition of chemistry that I've seen is the one by the biochemist who won the Nobel Prize for discovering the enzyme DNA polymerase. Arthur Kornberg. He probably fired the first shot of the biotechnology revolution. Kornberg called chemistry the lingua franca of the medical and biological sciences. So in fact, it is the underpinning of both biology and medicine. So it is important that one understands a little bit of chemistry. How does chemistry differ from biology? I don't think it really differs from it in any significant way. It differs from it only in length scales and in time scales and in the complexity of chemical interactions in biology. I have now put on this, taken from the internet, I've given you the source, all the way from a carbon-carbon bond and ethane up to bacteria and erythrocyte. You can see that the coronavirus, which is now dominating our discourse, lies right in the middle, somewhere between a ribosome and a mitochondria. The importance of chemistry is summarized in this cartoon. You have this man solving a crossword puzzle. He asks a question to the lady sitting next to him. What's a 9-letter word for biotechnology? The very clever lady, she immediately answers chemistry. And today, if you ask in what is the 9-letter word for material science, I would once again say it is chemistry. Now, one of the difficulties with chemistry is this, that in order to learn chemistry, one needs to learn structures. And chemical structures are really the alphabets of chemistry. In some sense, chemistry is like mathematics. If you're not comfortable with symbols and equations, which are the alphabets of mathematics, you're uncomfortable with it. So in order to learn chemistry and to learn mathematics, you need to learn a language first. But of course, chemistry is recognized by very important people. The Prime Minister, for example, said after the last general election that chemistry defeated arithmetic in the 2019 poll. He was right, because he had an intuitive understanding of what was meant by the chemistry between people. We'll come back to that in a moment. But I'm going to tell you a story now. At the beginning of the pandemic, when we were locked down, I read an article. The article was entitled, and it was in the magazine, The New Yorker, and it said, "Does hurting make us human?" It described the Scottish lady pictured on the bottom left of this slide, who, because of a strange combination of genetic quirks, a negative emotional range was enormously limited. She had a near inability to feel unhappy, to feel awful, and she had an expansive capacity for positive emotion. I thought this would interest neuroscientists. So when something bad happens, a brain immediately searches for a way to ameliorate the situation. It does not dwell on unhappiness. But what was even more important was her unusual emotional composition also was accompanied by a complete insensitivity to physical pain. It was then investigated after the physicians had discovered a unique phenotype. She was investigated by a powerful group of human geneticists, and they discovered that she had a mutation. And the mutation really led to a defect in an enzyme. This is why I emphasized enzymes at the beginning. Fatty acid amide hydrolase, which I've shown here. What does this enzyme do? It takes the molecule on the left, whose name, interestingly, is anandamide, and breaks it down into two components, arachidonic acid and ethanolamine. As I will show you in a little while, arachidonic acid is a product of lipid metabolism. And ethanolamine, of course, you all know, is a constituent of biological membranes and phosphatidylethylamine. It's also a constituent of the neurotransmitter acetylcholine, which is nothing but 3 methyl groups on the nitrogen and an acetyl group on the hydroxyl. If you put these two chemicals together, you make anandamide. So anandamide is something which is probably made in the brain, and it needs to be hydrolyzed. So this is her genetic defect. But now what I'm going to show you, I'm going to digress for a moment. I'm going to show you the long history of the chemical structure determination of the active constituent of marijuana and the understanding of its action on the brain. The story begins in 1838, when an Irishman in India, at that time we were under the British, investigated the medicinal uses of cannabis in India. Cannabis has been widely used, the cannabis plant extracts, hashish, and he began in 1838. It took all the way till the mid-1960s, 1964, for the structure determination of the active constituent. And from the 1960s to the 1990s, it was an understanding of what was the receptor in the brain and what was the endogenous molecule to be transitioned in our understanding of marijuana's effects from chemistry to molecular biology to cell biology. But our starting point, of course, was in human practice or in human cultures. Why is this important? Because today, of course, marijuana is again in the news. When important people are caught with hashish or marijuana, they are, of course, immediately arrested by the National Narcotics Bureau. But marijuana research has a very long history. As the most distinguished organic chemists of the first half of the 20th century, Roger Adams, summarized a lot of work that he did on marijuana in 1942. He was a very respectable scientist. He repeatedly risked his career and his position, both for his cannabis research and his political ideals, standing up to the forces of intolerance in a very paranoid age. Now it turns out that both intolerance and paranoia have never ever gone away from the time of Roger Adams. They keep surfacing. And it turns out that some of the most distinguished organic chemists of their time, Adams on the left, Alexander Todd on the right. Alexander Todd is the man who won the Nobel Prize for finding coenzyme A and so forth, and was also the PhD supervisor of Harpo bin Khorana, who was instrumental in introducing all the methods of nucleic acid synthesis and also in determining the genetic code. But the structure of the active principle of marijuana was worked out in Israel by the scientist who's pictured here, Raphael Mechoulam. He published this paper in the Journal of the American Chemical Society in 1960. And then he said sometime later that the endocannabinoid system is very important. Almost all illnesses we have are linked to it in some way or another, and that is very strange. So he then began to do research, which was very, very important. He asked the question, how is a plant product now acting on the brain? There must be receptors for it in the brain. But evolution has not placed receptors for tetrahydrocannabinol in the brain. There must be an endogenous molecule. With this argument, what he did was he began to work with pig brain, and from pig brain, 4.5 kilograms of pig brain, he isolated 0.5 milligram of a molecule, which now displaced in brain fractions, synaptosomal membrane fractions, displaced radio-labeled tetrahydrocannabinol, which meant it was binding to the same site. He then determined the structure and found arachidonyl ethanolamide, which he then called anandamide. Ananda, of course, is from Sanskrit, and he was asked then, why did you choose a word from Sanskrit instead of looking for a Hebrew name? Mechoulam then said, you may well be aware Jews are not very happy. We have a lot of words for being down and so on, but not so many words for extreme joy, which means since you are neuroscientists, you must be interested in emotion. And if you are interested in emotion, you will be happy to know that our ancestors were probably much happier than the ancestors of the Jews. Arachidonic acid itself is there in membrane stores, and you can have release of arachidonic acid, which can then be used to build up anandamide. This slide is a digression, and I'm going to come back to this because Anandya mentioned that I had done some work with Professor K.S. Krishnan. But when Professor K.S. Krishnan had begun to do the work, which I will describe a little bit later, he wanted to do some experiments on fish. By this time, I was in the administration of the institute, and I was at that time the chairman of an animal ethics committee. And in that committee, when this project was presented, one of the external members of the Animal Ethics Committee asked, do fish feel pain? Because you're going to inject fish with something, do fish feel pain? Of course, I had no answer to that question because I did not know anything about fish at that time. That possibly I'll have to ask Professor Krishnan. He then produced the article that you see down below, which says that worms and crayfish feel no pain. And there's an article back in 2005. The left bottom of the slide, you will see an article from 1979 from the New York Times, which says worms can feel pain. And then, of course, in 2005, scientists say worms cannot feel pain. Then in 2019, which was long after I had retired, I found this article which says there is ample evidence to demonstrate that fish experience pain. So you can see that scientists also change their views over time. And I guess neuroscientists change their views probably a little bit more often than other people. The arachidonic acid-related compounds, what we can now call glycosinoids, or C20 molecules, they're very important in inflammation and in pain perception. And if I show you this slide, arachidonic acid eventually goes to the prostaglandins, which are very important. And today, the entire class of anti-inflammatory molecules, painkillers, as we might sometimes call them in popular parlance, all of them are inhibitors of the cyclooxygenase reaction. But the arachidonic acid chemistry is very important because when you look at it, you will find that somewhere along a molecule like stearic acid is taken. And from stearic acid, oleic acid is made. Even if you're not a chemist, you can see that the hydrocarbon chain is extremely unreactive. And suddenly, in the middle of the hydrocarbon chain, 2 hydrogen atoms are removed to give you a double bond. I will just have a quotation here from Konrad Bloch, who won the Nobel Prize for his remarkable work on elucidating the biosynthesis of cholesterol. In 1969, Bloch reflected. He said, the stereospecific removal of hydrogen in the formation of oleate, although predictable on principle grounds, in chemistry, we believe that enzymes can do magical things, but they can do so much and no more. And this probably is one of their limits. I showed you that slide only to emphasize one point, that chemistry and biochemistry are important. Reflecting what would likely happen in the new century. And I used to read these articles, and Sidney Brenner wrote this in Trends in Biochemical Sciences in 2000. He says, I once made the remark that 2 things disappeared in 1990. One was communism, the other was biochemistry, and that only one of these should be allowed to come back. Of course, Brenner had not visited Kerala, I believe, and although Brenner would be very much warmly welcomed now in Bengal, which was the other state in which communism flourished. But biochemistry, of course, has flourished even through all of this, and Brenner recognized this by saying, we do not have to resurrect biochemistry. It will flourish because it provides the only experimental basis for the causal understanding of biological mechanisms. This is important for students to recognize. The underlying chemistry, the underlying biochemistry is what allows you to get a mechanistic understanding. Now I shall turn really to the only area where I can claim some sort of familiarity with areas which might border the neurosciences. But I make no claim to having entered these areas on my own. Professor K.S. Krishnan, who is pictured on the slide, was actually my first PhD student. When I joined the Molecular Biophysics unit in the end of December of 1973, I was less than 25 years old. I had no idea of what research meant, and I had no idea what research could be done in India. One of the first persons I met was Professor Krishnan. He was not a professor then, he was a PhD student, and he was actually looking for a PhD supervisor because D.N. Ramachandran had taken him into the department, and of course he hadn't been assigned a proper supervisor. On the first day that I met him, he was 3 years older than me. He looked at me and said, "You are now my PhD guide." So I would really consider K.S. Krishnan as my mentor. And so while I was officially his mentor, informally, we were both amateurs trying to learn what we wanted to do in future. But sometime in the 1990s, many years later, when he was at the Tata Institute of Fundamental Research, now as a professor interested in neurobiology, he came into my laboratory one day. I was still in Bangalore, and he came to my laboratory. He had a backpack, and he opened the backpack and turned the contents of the backpack He brought them, they look beautiful, but they don't smell very good. Are able to attack every membrane, receptor, and channel in the neuro, in the central nervous system, and therefore we should still look at the molecules. I asked him, "What will you do if I'm going to look at the molecules?" He said, "I'll go back to the seashore and collect more shells and bring them back and give the snails to you. I'll keep the shells." And so he had this wonderful collection of shells. Which debilize prey. They paralyze prey, so they act at the neuromuscular junction. Post-translational modification of biochemistry, you can summarize this area, many peptides and many enzymes. Professor Oliveira at the University of Utah, who really began this area of research, today calls it conotoxinomics, because it's very base word to add the suffix omics to any area of biological research. But generally what it means is this, that a very large number from the cone snail which will block potassium channels, sodium channels, calcium channels, the NMDA receptor, the GABA receptor, what have you, anything. Only one molecule has actually entered pharmaceutical practice, that is the conotide, which is used only for pain relief under extreme conditions, because it has to actually be given by a spinal injection. But what we really see here is diversity. This diversity is the cone snail makes a Q molecules. This is because it wants to immobilize diverse prey. How does it do it? Because these molecules now bind to receptors on the prey and paralyze them. The receptor is already there, but the prey now doesn't want to be eaten by the snail. So over time, the prey evolves and the predator evolves. So both receptors and toxins now evolve. hypothesis in ecology called the Red Queen hypothesis, and that can be very well used here. It was introduced by this ecologist, Van Bellen, who introduced this hypothesis using a little story from Through the Looking Glass, Lewis Carroll's famous sequel to Alice in Wonderland. In this sequel, Alice meets the Red Queen, and both Alice and the Red Queen are on a checkerboard, a chessboard, black and white squares alternating. And on those squares, in some places, there are bishops, knights, rooks, and pawns. Alice sees this and she tells the Red Queen, let's play the game. So the Queen takes her and they both run around. After running around for some time, Alice, who's a very good student, makes an observation. For a long time, but we don't seem to have gone anywhere. We're still in the same place. The Red Queen, who's an outstanding professor, now gives the interpretation. He says, now here you see, that is the looking glass world, it takes all the running you can do to keep in the same place. This is a rather profound statement, because sometimes in real life you run around a lot, but you remain in the same place. This often happens in research. What does this mean in the context of predator toxins and prey receptors? It means that they evolve over time. But it also tells us same. You can make new molecules by simply the atoms. You have enormous diversity, but the interactions between the molecules will still remain the same. They will evolve. And over evolutionary timescales, there will be this equilibrium where the predator and the prey then live reasonably comfortably in coexistence. Today, when we are looking and viewing the pandemic, you will see that these sorts of idea pandemic, the coronavirus pandemic evolved. But I'll come back to my title. Behavior and insertion mutation. I've changed it and behavior. This insertion mutation is the orbit of the coronavirus has an insertion mutation. If you talk about chemicals and behavior, we can go back to classical work, the work of Andrew Charlie and Roger Gilliman, which gave us all the hormones, all the hypothalamic hormones. And the hypothalamic hormones were the ones, hypothalamus there at the base of the brain, now of course get signals from the brain, and then it makes molecules which it secretes. These molecules in turn act on the pituitary gland, which then secretes the pituitary hormones. So the hormones which are very important, which come from the anterior lobe of the pituitary, are the adrenocorticotropic hormones. They are GnRH, somatostatin. But from the posterior lobe are oxytocin and vasopressin. And I'm going to tell you a little bit about this. The story is very interesting here. The characterization of the very first molecule, thyrotropin-releasing hormone, appeared in 1970. And in 1970, the techniques of chemistry were primitive. Mass spectrometry was in its primitive stages, and NMR spectroscopy was in its primitive stages. Yet the thyrotropin-releasing hormone structure of all peptide blocked at both ends, pyroglutamic acid on the left, a post-translational modification, C-terminal amidation on the right, modification, something that we recognize as a common feature of neuropeptides today, were first found there. You must also, for those of you who are students, you must realize the realities of scale. Heroic experiments. They used 25 kilograms of sheep hypothalamus. You can imagine how much weight of a hypothalamus is. And from all of that, by all these painful procedures, they got 1 milligram of the material whose structure they needed to determine. A few years later, the birth of neuropeptides. The enkephalins were discovered. The enkephalins and the endorphins are endogenous ligands for the opioid receptor, much like anandamide is the endogenous ligand for the cannabinoid receptor. I've shown you the structures of methionine, enkephalin, and morphine once again on the right bottom of the slide. On the left is just an image that I've picked up of a membrane receptor, one of the opioid receptors. Large receptors recognize these small molecules. There is almost no structural similarity between these molecules, although people have looked for it for a long time. Yet these have driven the field of neuropeptides. They were found again by the early techniques of fast atom bombardment mass spectrometry, which had just arrived, and the structure determinations are a famous episode in structure determination. Eventually, I come to the molecule with which I wanted to end this lecture, that is oxytocin. Oxytocin and vasopressin are hormones which are secreted by the pituitary gland, makes them, stores them in neurosecretory granules, and then in response to external stimuli, it now secretes them. Oxytocin and vasopressin have the structures that I show you here. I've gone to the conventional way of representing peptide structures. I'm a peptide chemist, and I use the one-letter abbreviation. It's somewhat more comfortable than using the chemical structure. Now, oxytocin is made in our brains. It's a widely used molecule today in medical practice. It's used for induction of uterine contraction for pregnant women during labor. Oxytocin works on smooth muscle, induces smooth muscle contraction. Vasopressin is very important as a diuretic, and therefore these are two molecules which contain important physiological functions in the body. On the other hand, which I've been investigating, make molecules just like this. These are analogs which are called the conopressins. So one of our questions was, what do the conopressins do? Insulin to cause hypoglycemia in a prey. It also probably weaponizes conopressin. That's not my subject here. I just want to show you a picture of Vincent Duvigneau, who determined the chemical structure of oxytocin. Oxytocin is a historic molecule because it shows you the transition between the methods of organic chemistry and the methods of biochemistry. 1 year before Sanger reported the synthesis of insulin, Duvigneau reported the first peptide hormone structure for which he quickly received the Nobel Prize. That was oxytocin and vasopressin. He then synthesized the molecule and showed that it had the hormonal activity of oxytocin. Why do I show you this today? This is because oxytocin is driving a new field of neuroscience. This is a cartoon which I picked up from the literature, Chemistry of Love. And there you have oxytocin. There you find the boy who's waiting as a suitor. Girl is busy drawing the structure of oxytocin, the chemical structure of oxytocin on the board. What does oxytocin really do? This really is my last technical slide. I've taken this from the very recent literature so that you can all read it. I've already given you a reference to the field of social neuroscience in an earlier slide. This has a very provocative title, The Social Neuroscience of Music, Understanding Social Brain Through Human Song. And it's just appeared in the American Psychologist. And these authors provide this remarkable illustration. You can imagine how difficult it is to prepare such illustrations. But I taught in a lecture that I gave day of the National Brain Research Center. I must at least have something that shows you the brain, even though I'm not a neuroscientist. But what has been so far established is oxytocin is produced in the brain. Oxytocin has dramatic effects on brain function, and that there are oxytocinogenetic pathways in the brain. And neurons which produce oxytocin now communicate with other centers of the brain. I will only tell you what these authors conclude, because I'm sure most of if you will be able to read this much better than I do. See an abbreviation like PFC, I have to go back and read that it's the prefrontal cortex and then go somewhere else and see where is the prefrontal cortex in the brain. I don't really understand these things. What these authors really say here is this. They say, we conclude that the COVID-19 pandemic could be a starting point for an improved understanding of the relationship between music and the social brain. It will be the starting point for an understanding, I believe, of many things. Understanding a starting point of how we perceive poetry when we read it loudly, and so on. In a time when people across the globe have been unable to meet in person, they have found a way to meet in the music. But long, long ago, in the 19th century, 1871, and this is quoted in this paper, because this paper really starts with a quotation from Steven Pinker, in which he dismissed in many ways music. He just called it something like dessert on the real functions of the brain. Darwin, in The Descent of Man, had noted that oxytocin has become a very important molecule. In fact, it has become also a molecule of abuse. A year or two ago, you will find that the Indian government had also worried about how oxytocin should be sold in pharmacies. And sometimes when restrictions were put, the gynecologists and obstetricians are unable now to source oxytocin for women in labor. So it is important to realize, and that is really the point of my lecture, that there is a very strong connection between chemistry and biology, and that there is a very strong connection between chemistry, biochemistry, and neuroscience. In concluding this lecture, all I need to do is to pay a tribute and an acknowledgement to the two institutions which have sheltered me throughout much of my professional life. The Indian Institute of Science on the top left, and over 4 decades I worked there, and the National Center for Biological Sciences, which has provided me a home in my retirement years. Thank you very much.
Thank you. Thank you. So, excellent. So now we'll have a question and answer session. Dr. Saurabh Banerjee will moderate this session. He's collecting the questions. Sir.
Bhavani, do you have some questions in the YouTube?
No, unfortunately, no questions in the YouTube.
Okay. So, anybody has any question, you can give it to me. Okay. So, Professor Balaram?
Yes?
Yeah. So there is a very interesting question. Do plants have emotions since they have so many neurotransmitters?
I can only say this, that Jagdish Chandra Bose always thought so, and he always thought that plants responded to music. and he did some very, very interesting kinds of experiments. And I think one of the less well-recognized contributions of J.C. Bose are his contributions in plant physiology. He's so well recognized for his contributions in physics, but it is much less appreciated that really how we define response. You can see that the literature has great confusion even on whether fish feel pain. So changes with the decade. The absence of a centralized nervous system I suspect, does not mean very much.
Next question.
Yeah. So the next question is, how will changes in the stereochemistry of chemical compounds be able to keep up with the plasticity of the brain?
I don't think you really need to change stereochemistry because nature's chemistry in some ways is limited. And what you do really is only to change sequences if they are peptides. And there's such a diversity of sequences that and flexibility, you say plasticity in the brain, I'll use the same word on the other side, the plasticity of peptide structure is enormous. Therefore, you can bring atoms into position by many, many different routes.
So I would like to ask one question. So how do the brain optimize neurotransmitter for any single cognitive function?
Well, this question I can't answer because I'm not a neuroscientist. I suspect this is a question that somebody in the audience should be able to answer, if it can be answered. I suspect there are a lot of questions in neuroscience which can't be answered.
Correct. So is there any other question? So there are two questions. Is pain subjective? That is the first question. And the second one is, do painkillers have uniform effect in all humans?
I must say here also, I do not know. Like, for example, I showed you the example. There are many people who are relatively insensitive to pain. In the example that I showed, it was a unique kind of genetic defect. But there have been other examples. In fact, one of them is something that Professor Krishnan and I looked at many years ago. There was a Pakistani family where there was a genetic defect reported in which it was a defect, I think, in one of the potassium channels. And the result of it is that the entire family was insensitive to pain. There are many ways in which pain insensitivity might be there. All people don't feel pain to the same extent. This is levels of tolerance of pain are different. There are other ways in which pain is alleviated. In the early days of neuropeptides, some remarkable stories emerged for the endorphins when they were discovered. Because it turns out that people had made observations that in battlefield injuries, for instance, wounded soldiers, they should feel incredible pain, but they're still able to not pass out. And this is largely because of the secretion of endogenous endorphins, which alleviate the pain. So there are evolutionary mechanisms which enable us to deal with extreme pain. C.H. Lee discovered at that time that camel endorphin was different from other endorphins, and camels have an extraordinary insensitivity to what I wouldn't say pain, but to physical discomfort, which are imposed by human beings on them.
Is there any more questions, Saurabh?
So yeah, another interesting question is
Yes.
why young PIs are not so interested to address questions in the areas of classical biochemistry.
Of me, except that I'm retired now. I'm certainly interested in questions in classical biochemistry. I know lots of people who are interested in classical biochemistry. Unfortunately, all of them are old. Sometimes it is a generational change. You know, I wanted to put a slide, but I have so many slides that I couldn't find it. I wanted to make it again, but Arthur Kornberg wrote an article in Biochemistry in the late 1980s, when molecular biology was actually flourishing, was reaching its peak. The molecular biology revolution following gene cloning had actually swept everything. He lamented in this essay that molecular biology, and I don't remember the quote exactly, in its tumultuous advance, has swept away all the bridges to biochemistry. This really meant that molecular biologists had forgotten much of classical biochemistry and the utility of classical biochemistry. Today, molecular biology and biochemistry are integrated into one another. There's no biochemistry without molecular biology. But today, what has happened is their own cell biology, driven by microscopy and imaging, dominates the field. If you look at it, you won't glowing. But the result is we are into visual imaging, and we are no longer working in certain kinds of as you are in the brain. Then, of course, these are important. I think in the paper that I showed you in the last slide, you do get a great deal of detail. And I think it's just an evolution of subjects, that's all.
Okay. So if there are no other questions, so please join me. Thank you, Professor Balaram, for his second very insightful presentation. And so we have a very small ceremony here. That it's a very tradition that on this Foundation Day lecture, we confer the Foundation Day medal to the speaker, and I would like to invite our director, Professor Padmanabhan, to come to the stage.
Thank you, Saurabh. Thank you, sir, again. This is our, I just want to say that I'm so happy and proud that speaking before you. So I was a student in ISC Bangalore and the SSU, and I visited your lab with Orindam, and I have so I'm still remembering that time, sir. It's my honor actually. So this is a sir. This would have been a great event if you would have physically that we generally give a medal to the distinguished speaker, and I will come to ISC and with a suitable time and definitely personally, and this occasion will happen there. Just thank you, sir. This is we give a medal, and this is the things we have been following for last many years. With this, where is the camera? Okay. Sir, it is all, everything is online, so we just want to show that this is being.
Thank you very much.
So please share. Yes.
Dr. Mandal, all I can say is this, that you can give me a medal virtually. You can't give me a cup of tea virtually.
Yes.
That's very
Yes.
sad, yeah.
Okay. So now goes back to Anindo. Anindo, please continue.
Yeah. So I would like to sincerely thank Professor Balaram on behalf of NBRC for taking time out from his busy schedule and to deliver this lecture. And we are really enlightened by this lecture, sir. And also, it is time for me to thank NBRC administration, finance, computer center, academic section for the support to support this event. Finally, I would like to thank all the students, faculty members, all the honorable guests, and everyone to be part of this event. And I would like to conclude this event here. Thank you all.
Should I? So just minimize it and that's it. I can come out of it, right?
You can leave the meeting.
Okay.
Or you can mute yourself.
No, it's okay. Leave the meeting.